Seth. How are you doing today? Mark just sneezed really loudly, just in case you missed us. So I guess this thing is recording. So last time we talked about non-tracheophytes, so plants that did not have trachea and they did not have any seeds, so they're very simple, like mosses and liverworts and hornworts. So today we're going to move up on the plant chain of life a little bit, and we're going to talk about seedless vascular plants. So these are plants that are still going to produce spores, but they're going to be taller because they actually do have trachea, which provides support. So we're going to start with division Lycopodiophyta, and we're going to talk about Selaginella and Lycopodium. And you can actually see Lycopodium coming up all over the forest right now, which is kind of exciting. So the princess pine. Has anyone ever seen princess pine before?
Is that
They stand about maybe that tall. Yeah, the first time I ever saw them, I was just like, baby pine trees. And then one day I was like, wait a minute. And I looked and they had a strobilus. We're going to talk about what a strobilus is and why it's important. So we're talking about things that look like that. And then we're going to move on to what they call the ferns and the fern allies. So division Pterophyta. Pteridophyta. They used to call it Pterophyta. So if you learned it as Pterophyta, that's fine. I won't mark you off, but according to our book, it's Pteridophyta. So we're going to look at order Silotales, order Aquasitales, and order Philotales.
Are you sure it's not like this is how they used to call it? Because this is an old textbook.
Yeah, it is a little bit older. But a lot of people still use this taxonomic classification. Okay. So if you recall, we talked about green algae as kind of being the ancestor to all of the plants. Do you remember why that is? Anybody? How to do with pigments? What two chlorophyll pigments do plants have?
A and B.
Yeah, chlorophyll A and B. So green algae, these were the first organisms to have both chlorophyll A and B. And then if you step up from the algae, you get rhizoids. So if you recall, for those of us who were here last time, those little liverworts, remember how many of those root-like structures they were producing? They're going crazy. So those are very primitive anchoring structures that actually do a little bit of absorption. And then the hornworts, as we get into the hornworts, they have stomata on their sporophyte. We talked about how that sporophyte, it does get off the ground a little bit more. It sticks up a little higher. And when you're away from the ground, you are more prone to drying out. So that was the first place that we saw stomata. So the bryophytes, the mosses, bless you. Everyone's sneezing today. Is that it?
I think so.
No more sneezes?
No.
So basically, the mosses, if you've walked outside, you've probably seen mosses on the ground. You've probably seen those little brown things that stick up. If you haven't noticed them, take a better look. Those are the sporophytes, and we talked about those a lot last time. And there were a couple of videos that we watched during lecture. I didn't record them for Seth or for you guys because it's just too hard to get all that to work. But just go into the lecture and click on, as it's in presentation mode, you'll see life cycle video. Just click on that, it'll take you to it. And you will be seeing similar videos probably today if we get to it. So once you get past the bryophytes, you notice that we get into lignin. Remember, when something is lignified, it's tough, right? So lignin makes things tough. Then you get xylem with tracheas, the dominant sporophyte, branch sporophyte, and rhizome. Okay, so we've gone from having rhizoids to rhizomes. And so all of these, remember, they do not have what's considered to be a true vascular system. They're basically just like little tubes that act like capillaries. So anyone ever played with capillary tubes in classes?
They're
Yeah.
a pain in the neck.
What?
They're a pain in the neck.
They are a pain in the neck. Do they still do that blood heparin testing in anatomy?
In bird phys we did.
You did it? Did anyone pass out? Yeah, I always had students pass out whenever they do that. I don't know why. But anyway, that's how these guys got water. But these guys that we're going to talk about from here on out, they actually have what's considered to be a true vascular system, and they have tracheas within their xylem. So here's our first group, the Lycopodiopida, with Lycopodium and Selaginella. I have a whole bunch of Selaginella in my office right now. It's just taken off for some reason. They're very pretty, and if anyone's interested in having their own little jar garden, I have plenty of propagules if you want to have a couple liverworts and maybe some Selaginella in a jar, maybe some mosses. It's a big thing in Japan right now, actually, fairy gardens. So. Okay, Pteridophyta, Equisetalis, Silotales, Philatales. Okay, so we're going to start our discussion here at the Lycopodiopida, and then we'll continue with the Pteridophyta. Okay, so let's talk about the general features of the ferns and the fern allies. Okay, remember how last time we talked about how all that fluffy green stuff, that was all gametophyte tissue in the mosses, and the sporophyte grew out of that gametophyte tissue in mosses. Now what you have is you have a dominant sporophyte. So the sporophyte is no longer dependent on the gametophyte, and now the life cycle, the dominant part is the sporophyte. So we are a sporophyte dominant organism, right? The sporophyte is branched. So if you recall those little phyllids on the mosses, they weren't really branched, were they? It was just a single stalk kind of coming up with little phyllids coming out the outside. Ferns are actually going to have a branched sporophyte, and they're going to be mostly homosporous. So remember when we were starting to talk about plant taxonomy of the land plants, how we talked about homospory and heterospory? And heterospory is important because that's going to lead to pollen and eggs contained within ovules, okay? We haven't gotten there yet with this group. They have spores that basically, they look all the same, but they're going to grow into gametophyte tissue. And then they have true vascular tissue with lignin, and they have tracheids in their xylem. And they have a rhizome. Remember how we talked about plant organ modifications and rhizomes? They were kind of a lateral root that grew underground, and that they could produce new plantlets off of them. Do you guys remember that? So kind of a similar thing. It has adventitious roots, and those roots, that means that they can get pretty much everywhere, and you can get more ferns from them. And many have what's known as a strobilus. A strobilus is like a cone, and we'll talk about that a little bit later, but it's a really neat structure that evolved in plants to allow them to produce as many spores as possible per unit area. And then they have true leaves, microphylls or megaphylls. So last time when we looked at phyllids, the phyllids in mosses, they do not have true vascular tissue, right? In order to have a leaf, you need to have at least one vein. And a microphyll has one vein, and a megaphyll has more than one vein. Okay, so let's talk about when they dominated. So they dominated during the Devonian period. So this is what the plants used to look like. Could you imagine walking around in a place like that? I mean, I know people who have gardens like this. I have a bunch of botany friends, and they're like, yeah, look at my Devonian garden. And they weed out anything that's higher up. But, you know, we're an interesting group. But these are the wisp ferns here. We have some Lycopodium. And this guy right here, I'm not really sure what they were going for there. I think that one's probably some sort of a lycopod. Okay, so by the time we get to the Carboniferous period, you have not just those earlier forms of the ferns and the fern allies. Now you have true ferns. And I want you to check something out. These guys, they're not trees. They're tree ferns. And I don't know if you guys have ever seen tree ferns. They don't do so well out here, but at UC Santa Barbara, we had this whole area. It was a courtyard. It was kind of darker, and they had all these tree ferns. And they can get, I don't know, 15 to 20 feet tall, at least now. Back in the day, they got really tall. So even though this looks like it should be considered wood, is it really wood? No, remember how we talked about secondary growth and xylem? Secondary xylem making up wood. So this was when it was very warm and there was swampy lowlands. And actually, incidentally, the Devonian period, because this one was also very warm, this is when you started to get fish coming out of the water, because the water got warm and the oxygen contents decreased. And by the time you get to this Carboniferous period, you have quite a few amphibians walking around. Some of them were rather big. You don't want to mess with them. All right, so let's look at their phylogeny a little bit. So we kind of group them into the lycophytes. You can see that they have all of these different characteristics. But we also group them into euphylophytes, monolophytes, and lignophytes. Okay? So you can see that you have this branch here at about 420 million years ago, where you have the vascular tissue and then tracheids that are strongly lignified. This is a major branching point right here. Okay? Transverse dehiscence of sporangia, meaning that it's going to be basically released that way. This indicates all these groups right here. But notice how there is secondary xylem in some of these, right? So that means that some of them might actually have what you could call true wood. Is it going to be exactly the same type of wood that we looked at before? No. So there's an argument there. But notice that they have microphyls over here, and they have heterostyle. So heterostyle, this has to do with having different sized males and females, okay? And then here we can see overtopping branches. So you actually have branches that come out now, okay? Instead of just going straight up. And this is where you get into what we call basically the ferns. And here you have the lignophytes that leads up to seed plants. Okay, so seed plants, we find them all the way over that way. Okay, so many of these seedless vascular plants, they have what's known as a strobilus. So this is a strobilus right here. It's a cone-like structure, and it consists of a sporangium. The sporangium is the structure that produces the spores. Okay. So that sporangium, what process does it use to produce the spores? Is it producing the spores through mitosis or meiosis? You guys need to remember those life cycles. Yes, meiosis, okay? So they're producing their spores by meiosis, but they're producing their gametes by mitosis. Okay, they do it differently than we do. Yeah, but you had a 50-50 chance, right? So they're born on these sporophylls. So sporophyll literally means that it's carrying that sporangium. They also have this really weird arrangement of vascular tissue. So there's this arrangement of protosteels versus siphonosteels. So remember, we looked at a plant that had siphonosteels before. Do you guys remember what major class of plant that was that had siphonosteels? I'll give you a hint. It has to do with the number cotyledons. So, all right, you know, Erica she's trying, you know, and she has a 50-50 chance. So the monocots, they do, it's all right. I'm really happy that you're trying. So the monocots had this siphonostele structure. But before there was a siphonostele, there was this structure called a protostele. And that protostele had this arrangement. And there's several different types of protostele. So we have what's known as a haplostele, actinostele, and plectostele. So haplostele. So this is very basic. It's a cylindrical core, and it's looked at the roots of both seedless and seed-producing vascular plants. Okay, so we see this in the roots of both of these groups. So seedless and seed-producing vascular plants. So remember that that haplostele, it may be divided into branches, right? And those might be a tetrarch or a triarch if the little arms go off. Now the actinostele, so it's lobed or star-shaped, so it's rare. So basically it's rare in living plants, but it's found in horizontal underground stems of spore-producing whisk ferns. So the whisk ferns, they do have this structure. But this structure used to be very common. A lot of the living plants had this, but they went extinct. And then the plectostele. So this is when you have interconnected plates or walls surrounded by a matrix of phloem tissue. So check it out. So that red stuff is xylem, and the blue stuff is phloem, right? So in all these cases, the xylem is more towards the middle and the phloem is more towards the outside. But in this one, it's kind of weird. The plectostele, it's kind of like a grating in your bathtub or something. Okay, but there's discontinuous siphonosteles too. So selenostele. Okay, so we can call eusteles. So monocots and dicots, they produce something called a eustele. So they're arranged in rings surrounding the pith, or they may be scattered, like the monocots. So we call that a eustele. But we also have these other ones, the selenostele and the dictyostele, which are kind of weird. So we can identify the groups by their steles. So if you were to take a cross-section of whisk fern, you would see this. A lycopod or Lycopodiopida, you would see that. And then the horsetail, you would see something that looks like this. Okay, what does this look like here? What kind of stele is this? It's kind of brown and it has this little.
And it just looks like a
Siphonosteal, exactly. So Erica just rocked it right now. How about that one? It's a, what's that one? Actinostele. So the Silotales has an actinostele. How about the Lycopodiopida? What do we call that one? Plectostele. So the way I remember plectostele, it kind of looks like a mouth with teeth, or like a vampire-looking thing. If you've ever seen a plecostomus in an aquarium, they kind of suck on stuff. It's a stupid way to remember it, but that's how I remember it. And then you have the Phyllocales. If you were to take a cross-section of ferns, you would see these weirdos. Okay, so which one was this one on the top here, above the Phyllocales? Dictyo. How about this one? Huh? Selenostele. Okay, so let's talk some more about division Lycopodiopida. So we've got this really neat separation between the lycopods and Selaginella. So all of them have a strobilus, a plectostele, and they have microphyll. So they actually have a true leaf with a single vein that goes through it. But there's this neat division where Lycopodium, it's homosperous, but this split here, Selaginella, it's heterosperous. So you have large spores and small spores. And they have what's known as an endosporic male gametophyte. So that means that you're actually going to have a male gametophyte that's different than the female gametophyte. So we're going to talk about Lycopodium first. So this is an example of Lycopodium that you might see growing in the forest floor. There's a few other examples. Here's a close-up look at Lycopodium at a strobilus. And here, if you look in the sporangium, you'll notice that there's spores in there. Produced by what process? So produced by meiosis. and they're borne by a sporophyll, they're all the same size, right? So what do we call that? When spores are all the same size? Homospore, okay? So this is a homosporous plant. Okay, so let's look at a single sporophyll. We have the lycopodium, or sorry, the sporangium on the inside and sort of the sporophyll subtending that sporangium. Okay, and here's an actual gametophyte. So we have a foot here attaching to a shoot and a rhizome, and we have this large gametophyte coming off. So where did fertilization occur on this plant? What do you guys think? Where did fertilization occur in the mosses? Yeah, in the shoot. What structure in the shoot? It bears female gametophytes.
Archegonia.
Yeah, so archegonia, right? A complete and specific term for where fertilization occurs would be the archegonia. And we have this flat gametophyte growing out. So we have all these strobili, they're sort of interspersed with lichens. And lichens are really cool in that there's symbiosis between fungus and algae. And this is, these are strobili with microphyllous sporophylls. So here we can see our little microphyll leaves, and within those microphyll leaves, we have the sporangia on top of each of those microphyllous leaves, at least towards the top. So here's another sporangium sporophyll. So this is a microphyllous leaf here. Okay, and they're highly combustible, actually. I have some lycopods that are producing spores right now, and they're not quite ready yet. I don't know if I brought them in class and lit them on fire if I would get in trouble.
Probably.
Dr. Clark popped a balloon.
Really? Well, maybe I'll bring one and just don't tell anybody. Okay. Yeah, because it is really exciting. So they used to use it for flash photography. So if you've seen those old timey videos where they had the, you know, you think it might be gunpowder or something. It's actually a combination of combustible things and primarily lycopodium spores. And they use it in pyrotechnic displays because it's so explosive.
Yeah, it's fun. We had that in AP chemistry. We had about
Oh, yeah?
a few grams of it. It's pretty cool.
Mm-hmm. Although I think it would take a lot of time and energy to harvest that many of them. So I've tried to harvest them before. It's not so easy. But why are they flammable? Is it an evolutionary adaptation?
Probably not.
No. So here's another example of some strobili that are growing off the top here. Okay, let's go through the lycopodium life cycle. Oh, I thought I broke it up into littler pictures. So let's start, let's see, where should we start? Let's start with our strobilus here. Okay. So our strobilus is going to contain sporangia borne on sporophylls, and through meiosis is going to produce spores. They're all the same size, so this is homosporous. They germinate and they become a young gametophyte, just kind of this flat paddle looking structure that starts to, whoops, not used to this computer. Hold on, I'm looking the wrong way. There we go. All right. So it's this little paddle shape right here. It's going to grow into a larger structure that's going to bear archegonia, which produces what type of gamete? Egg. And then antheridia?
Sperm.
Sperm. And some of them have a symbiotic fungus growing with their gametophyte, which is kind of cool. So, you know, the role of a fungus sometimes. Do we remember talking about mycorrhizae? What did mycorrhizae do for roots? Someone other than Mark. So what does increasing surface area volume do with respect to absorption? Increases absorption. So this is kind of the first time we see this in a land plant. And if we look closely, here's our immature archegonium with a developing egg and an immature, trying to get this thing to show, immature antheridium that's going to produce sperm. And it's actually, the sperm are fun because they're kind of double flagellated and they don't swim in a very directed motion. They kind of,
Tumble.
yeah, they tumble and switch. So they find their way into the archegonium and fertilize the egg. So do we remember how it was in the mosses that the sperm actually got to the archegonium?
There was like a chemical that was produced by it.
Yeah, so there's a sperm attractant, and in this case there's also a sperm attractant. Otherwise they would just be kind of like floating around and they'd never get there. So after fertilization we have a young embryo that's going to have this very primitive looking foot structure. We still have gametophytic tissue surrounding it. A root starts to form, and here's our shoot apex, shoot apex and the first leaf. So not as different as one of the embryos we were looking at before in a seed, right? But is it completely protected? Not completely, but it's better protected than other structures, right? Because it's actually partially covered. So this is what happens. The gametophyte stays there, but you're going to have this really interesting rhizome structure and adventitious roots that are going to produce multiple stalks per gametophyte. So here we have tiny leaves or microphyls and what's called an aerial stem with strobili coming off the top, and it's within those strobili again that you have the sporangium, which is going to produce the spores via meiosis. So this should look somewhat familiar. We're kind of building on previous life cycles that we've already talked about, right? So there's some themes that continue where you have the archegonia and the antheridia within the gametophyte tissue, and that it's them that produces the eggs and sperm, comes together to form the zygote. And then through meiosis, that sporophyte, once it's mature, is going to produce spores. So let's talk about Selaginella, which it's in division Lycopodiophyta, but it looks almost nothing like its sister group, Lycopodium. So here's an example of some Selaginella, and there's a number of different species. So here's square strobili. So these lighter tips, right here, these are the actual strobili. If you were looking at this thing and didn't know what you were looking for, you probably would just say, oh, you know, those are new developing leaves or something. They're not. Some of them we call resurrection ferns. Have you guys ever seen the resurrection plant? There's one in, it's floating around in the intro bio lab. And this plant can basically dry up and look dead, but then you add water and it'll start to unfold and it'll photosynthesize again. So you can actually buy yourself one that they've pulled up and made into a resurrection type plant. Okay, so very similar life cycle here. But, I mean, it does everything the same one did, right? But now we have this microsporangium versus megasporangium. So microsporangium is going to produce the small spores, and the megasporangium is going to produce the large spores. Okay. So if we follow the microspore, what we're going to get is spermatogenous tissue. So the small spores are actually going to be producing the sperm, okay, and the sperm is going to find the egg here. And then our megaspores, we have a gametophyte protruding from that megaspore wall, and the archegonium is located on that megaspore that's going to become fertilized. And then we have what's called a megagametophyte. Okay, so that megagametophyte, that's essentially the female gametophyte, right? The male gametophyte, basically, all it did is produce sperm. That's it. So we have all the same structures here as we did before, but if you notice, you have a megaspore wall, you have the first leaves coming up. They look a lot more mature, don't they? They're a lot more defined, and then the root coming down, pouring into this leafy structure which produces these strobili that through meiosis are going to produce those two different types of spores. So homospore or heterospore?
Heterospore.
Yeah, heterospore. And do you kind of see how the microspores and the megaspores have taken on male and female roles? So instead of just having an antheridia and an archegonia, now you have these separate, more separate gametophytes from one another. And so here's a closer view of the microspores. So they look kind of pokey. And then we have the megaspores. Notice how they're both haploid because they were produced by meiosis. So if you were to see something that looked like that, you should know that you're seeing Selaginella. And I know that the 110 folks, we did see, I think there was one slide of Selaginella floating around, and maybe one slide of Lycopodium. Okay, so let's look at the endosporic microgametophytes. So here we have spermatogenous tissue, and within that spermatogenous tissue, you have the rupture and release of that sperm. Okay, whereas the female, all she does is kind of split open a little bit, and when she splits open, her archegonia are going to become available, and that's when she's going to get fertilized. Okay, so notice how that process takes place. And then you have, through meiosis, the generation of those different types of spores. So here's a slide. You can see that if you see a strobilus, and that strobilus has sporangia, and those sporangia contain small spores and large spores, you're going to know immediately that you're looking at a heterosporous plant, and that heterosporous plant has started to take on real female and male roles. And of course, the supporting structure, what do we call it again? Yeah, sporophyll. Okay, so yeah, so the whole thing is the strobilus. But now that we're looking at megaspores and microspores, now it becomes even more annoying with terminology. So now it's microsporophylls and megasporophylls. If you were to look at this thing from the outside, would you know which one is a microspore or a microsporophyll and a megasporophyll? Not necessarily. So you'd have to know something about what's inside to make that much of a distinction. Okay, so what we call these in botanical language is microphyllis, microsporophylls, and microphyllis megasporophylls. So remember how we looked at leafy sporophylls. So instead of just having that little tiny pokey, it's almost like a sepal or something. It's not that defined, to having almost an entire leaf. So here's an example of Selaginella. So this is what they look like on the outside, and that's what they look like on the inside. Here's some microspores and a megaspore. So hopefully you'd be able to determine which is which. So here's another Selaginella producing a larger strobilus. Okay. So if you look at them really close, in some species you can actually tell the difference between the megasporophylls and the microsporophylls, because you can tell from looking at them that you have a megasporangium versus a microsporangium. This is not always easy to see. But see how the female one is sort of bulging, and the males are just kind of round. But again, you'd have to really look closely. So again, we looked at a couple different plants. We looked at Selaginella, which is Heterosporus, and they say that the strobilus is square in cross-section. I guess so. I guess it's semi-square, and you have a unisexual gametophyte, whereas in Lycopodium, it's homosporous, it's round, and has a bisexual gametophyte. So bisexual in this one, right, if we're looking at a homosporous plant, it would have to have male and female reproductive structures on the same thing. But if we're looking at a plant that you have heterospory going on, the male and the female spores, they're taking the roles, so they're each doing their own thing. Does that make sense? Okay. So there's a lot of argument still, not just in our textbook, which is a little bit older, but still we're arguing about exactly what the evolutionary relationship is among our wisp fern and horsetails and true ferns. So I'm going to be presenting what's most commonly believed and most commonly presented about these guys. So the wisp ferns, they look like that. So if you were to turn them upside down, you can use them as a whisk broom. The Equisetum or Order Equisetalis, these are horsetails. You guys may have seen these around ponds and places. They're going to have a very distinct strobilus coming out the top. And then Phyllocales, the common fern. So what we're looking at here is a fern gametophyte. And this fern gametophyte actually has Archegonia and Antheridia. So Mark has been looking at the growth of his fern gametophytes. They haven't quite gotten to this prothallus stage, but maybe on Thursday, could I borrow a dish of them for us to look at?
Yeah, sure.
Okay, because that would be cool. All right, so this Psilotum, which is in the Psilotales, the stems are what's photosynthetic. Okay. So it has photosynthetic stems and has underground rhizomes. Here's a close-up view. And here's the life cycle of Psilotum. So let's start with the sporangia. So as the sporangia start to grow, they start to produce what's called spore mother cells. So spore mother cells are going to produce spores that germinate, and they become a young gametophyte with Antheridium and Archegonium on it. And then as they mature, you can see that there's a mature egg, and the spermatogenous tissue is going to become sperm that are then released and fertilize the egg and the Archegonium to produce a zygote, a young embryo. And then you have this gametophyte containing the embryo, and you have something called a calyptra, young sporophyte, and a foot. And from the shoot apex, you're going to get this aerial stem, and then you have a rhizome extending down. This is a bisexual gametophyte in Psilotum. So you can kind of see, you can see these Antheridia pretty well. Okay, here's another close-up shoot of sporangia and the photosynthetic stems. So another close-up view. And here, what we're seeing is they're starting to release their spores here. And I need to get some specimens of wisp ferns in our greenhouse. I don't have any yet. They're really hard to find. If you just happen to be walking around somewhere and find one of these, let me know. Or, you know, if it's not somewhere you're going to get in trouble, dig it up. Okay, bring it to me. Because I've never seen this dehiscence of spores. It's on my bucket list. I know, I have a strange bucket list. So here's our horsetail. So division Pterophyta, order Equisetalis. Let's see, I did something there. I accidentally wrote Pterophyta instead of Pteridophyta. So I'm not going to dock you if you use either one. So I just discovered that I myself just made that error. So they were weird. They were so weird, they used to have their own division, so division Sphenophyta or Equicetophyta. But people are still arguing about exactly how they're related to other things. Because they are so weird. There's only one genus and only 15 species left of these guys. So they have whorled photosynthetic stems. And this is their strobilus. So if you see something that looks kind of like a horsetail, not all the horsetails have those stems coming out around the outside. Some of them are just the bare stalk with that strobilus on the top. And what you'll see is that each one of these things right here, the sporangia are on the underside of what's called a sporangiophore. So you have to look at them really closely to see that structure. This is a larger, or a zoomed out view. So you can see multiple sporangia. And I want you to see something here. These are the leaves. These little reduced brown looking things. Isn't that weird? So what we're seeing mostly are the photosynthetic stems. Yeah, Mark, did you have a question? Just stretching. Okay, so they have siphonosteles. And here's their gametophyte. So they have antheridia and archegonia and rhizoids. They're a little bit branchier. But they're covered in something called hygroscopic elaters. And these hygroscopic elaters are going to help them be dehisced or spread in the environment. They look like coiled springs, right? So here's an elater. So here we can see the gametophyte. Here we're looking at the stalk, and these are actually photosynthetic stems, and these brown things are the reduced leaves. So these are more stems coming out. Isn't that weird? So scientists have been scratching their heads about these guys, because you would look at that and think, oh, it's some sort of a modified leaf, right? No, it's a stem. Another strobilus. And a lot of them have silica, so sand-like substances. So the Native Americans, whenever they could find these, they would use them to scour their pots. So sometimes people would call them scouring rushes. Well, they're not rushes. So I'll have to find some rushes and sedges. Have you guys ever heard the little rhyme to remember the difference? So sedges have edges and rushes are round and grasses have joints all the way to the ground. So if you happen to be in an area where you find those species, in fact, maybe we do have something that's considered a sedge. I'll have to look at that. So if we look at their sporangiophores, they have this really interesting strobilus that is consisting of those sporangiophores that look like little umbrellas. They have this whorled stem arrangement, so remember that those are stems, right? They're not leaves. And they have elaters, which act like springs to release the spores. They have reduced leaves that don't do anything, and they have photosynthetic stems that do all the work. And again, they have silica. They're homosporous, so they only have the same structure of spores. Okay, so let's move on to the Phyllochales, the common ferns, which I think are probably the most charismatic of the different types of plants we've been looking at. So ferns are complicated. They produce selenosteles and dictyosteles. So in these selenosteles and dictyosteles, they have a central core of pith surrounded by a cylinder of vascular tissue, and the xylem, phloem surrounds the xylem. Okay, so notice that in these selenosteles and dictyostele, you have an incomplete cylinder. Weird, huh? Okay, so it's incomplete, and part of the vascular tissue branches off. And this is to allow it to provide nutrients to the leaf. So that's why it's broken right here. But it's really weird. Okay. So these are some other members of Pteridophyta that we're not going to explore. The maritoid ferns, maritoids, heterosporous water ferns, great ferns, adder's tongues, and allies. So here's Ophioglossalides. And I think we have one kind of like that, rattlesnake fern. Do you remember, Erica, that day we went out into the field and we found, yeah, that was rattlesnake fern. It looked a little bit more like that. I actually have some spores somewhere that I want to germinate. It's a very strange plant. So how long have they been here? They've been here since the Carboniferous. And there's all kinds of different sizes. You go from these really small water ferns to very large tree ferns. And water is still necessary. So everything that we've looked at so far, in order for those sperm to get to the egg in the archegonium, they have to be swimming in water. Sperm can't swim through the air. That's why pollen was invented. Not really invented, evolved, right? All right, so sporophyte's dominant. So the leafy fronds that you see on a fern, these are the dominant sporophyte tissue. And they have fronds and rhizomes. So the fronds, each one of the fronds that comes off is considered to be one megaphyll, and then there's a bunch of rhizomes in the bottom, so there's no true roots. And most of them have selenostellic or dictyostelic rhizomes. So those rhizomes have the same vascular structure as the stems. Okay, so generally they're homosporous, and the fronds are megaphyll. So a microphyll has one vein, a megaphyll has more than one vein. And they have sori, and they have an annulus, and we'll look at these. And many of them have indusia. So if we look at the frond structure, what we have is a central rachis, and then we have pinna coming off the side. Here's the rhizome, and adventitious roots coming off. So here's a frond with pinnae. And a lot of them, when they grow, they have fiddleheads. And some species, you can eat the fiddleheads. I don't know which ones are edible out here yet, but I will find out. Don't worry, I'll look it up before I eat them. They have something called circinate vernation. Okay, so circinate vernation describes what they're doing right here. Fiddlehead. And here we've got some sori and some aphids, actually. So this is what the sori look like. And if you were to look at a fern, kind of look on the underside, you might see button candies along the underside. Have you guys all had button candies before? Anyone not had them? Okay, we're gonna have to basically fix that. I mean, it's just like an experience eating button candies. I don't know why. What? Button candies?
Yeah, what's that?
Okay, yeah, I have to buy some and bring them in.
No, it's fun. It's like, what are they? Just describe it.
They're like little bits of flavored sugar balls stuck to wax paper, essentially.
Oh, yeah,
I'll have to look that up.
Yeah, no, I mean, it's just one of those childhood experiences that I need to make sure that you have.
So they're, we call them button caps.
Button candies.
Button candy. Want to write that down?
Okay, so these sori are actually clusters of sporangia. So it's kind of like, it's the same idea as Astrobolus, but it's different. So this is one of those. It's the same, but it's different things. So each one of these little dots in here is a sporangium that's producing spores on the inside. And since they're homosporous, they're only producing one size. So here we have the fern leaf. Each one of these is a sporangium, which produces spores through the process of meiosis. And there's like this ribbed cap that you find on the sporangium. That ribbed cap is going to be responsible for flinging the spores away. And then you have, in many cases, edusium, which is this stalk and cover coming down. So anybody know what indusium in Latin means? Yeah.
Underwear.
It means underwear. So some botanist somewhere thought it was funny that, well, it's covering reproductive structures, right? So, I don't know. People are weird. So here's an indusiate fern. So if you see on those button candies, there's like a papery cover, then you know you're looking at an indusiate fern. So what's the ploidy level of the indusium? Diploid or haploid? The indusium is an outgrowth of what?
Haploid, right? And diploid.
Okay, so the spores in the sporangium, right, those are going to be haploid, but since this is sporophyte dominant, and this indusium is kind of growing out of that leaf tissue, then it has to be diploid. The ploidy of a spore, diploid, haploid,
Diploid.
haploid. Yeah, so spores produce the process of meiosis and is therefore haploid. Difference between a sorus and a sporangium. So what's the difference between a sorus and a sporangium? Okay, so each one of those little dots is a sporangium, right? So a sorus is a collection of sporangia. Okay, so sorus is a collection of sporangia. So here's another view of an indusiate fern. So you can see that indusium like an umbrella covering the sporangia. And this is how the spores are released from a fern. So you have the unhinging of that sporangium, and the annulus actually pops back and then flings them like a spring. So here we can see some uncoiling here happening. So the uncoiling and then spring, it's like a spring-loaded mechanism. So ferns, a site for sori? No? All right, here's an interesting collection of sori covered by indusia. But they're not always like round button candies. They may also be longer stalks. There's a lot of different structural diversity here. Sometimes they may cover the whole bottom of those different pinnae. Here's an example of a fern that's producing a whole heck of a lot of spores. So here we can see this leaf before production and this leaf after production. Here's an example where the sori are sort of lining the outside leaves. Okay, and here's the fern life cycle. I need to see how long this video is.
It's pretty long.
Is it? Is it like 20 minutes?
It's like 10, I think.
Because we have 10 minutes.
It might be longer, but
All right, Seth, so we decided we're going to watch the video next time. But just really quickly, I'm going to point out just a few differences in the fern life cycle. So we have, each of these is a sporangium. And then within that sporangium, they're producing spores through meiosis. And they're going to be thrown with the annulus, germinate, and they're going to become this mature gametophyte called a prothallus. And that prothallus contains archegonia towards the top of that little heart, antheridia towards the bottom. We have the same fertilization occurring where the egg in the archegonium is going to be fertilized by the sperm. But the ferns have this really weird looking sperm. And it's going to move like a corkscrew. And then it develops into the young embryo. We have a very similar looking embryo with a foot, root, and here you have that first leaf coming off until you have that mature megaphyll. But the gametophyte is always going to be sort of there, just under the ground, and sometimes it'll kind of shrivel up a little bit. You'd have to really dig up a fern and look at it carefully to try to find the gametophyte. It just kind of, it hangs out. Okay, so I think we're going to end here today, so don't forget to have your rough drafts by Thursday.